All right. Thanks so much, Lachelle, and thanks to everyone for being here. It's a true privilege to be able to work with Rein in Sarcoma, and thank you for your support of this event. I also want to acknowledge Abby Mackey, who's done a huge amount of work to make this event happen again this year. So thank you, Abby. Our guests today include sarcoma patients and survivors, medical professionals, and supporters of Rein in Sarcoma. We have a full and exciting agenda ahead of us today, and I'm delighted to introduce our esteemed speakers who are leading the way in sarcoma research and treatment. Our first speaker is Dr. Aru Narendram, professor in the Department of Pediatrics, Oncology, Biochemistry and Molecular Biology, and Physiology and Pharmacology at the University of Calgary. He will share his insights on the biology, mechanisms, and targetability of cancer-associated fibroblasts and rhabdomyosarcoma. His initial set of studies to develop novel treatments for sarcoma was recently published. Please join me in welcoming Dr. Narendram to the virtual stage. After he speaks, we'll also open up the floor to questions for him. So go ahead and get started. Now, I guess if you share, we can go ahead and start. Is that what he's going to do?
Yep.
Okay.
All right.
Hey, thank you ever so much for the invitation to come and talk to you. I'm so sorry I couldn't be there in person. My colleague there couldn't move that around in time, but I'm sure I'll make it to the next one.
Okay. Sorry, Dr. Narendran, it's not showing in our in-person here, so let's just
okay.
Give us 1 second here. On the virtual screen, but not in the room here. Okay, got it. Yeah.
Okay. So I will talk about a project that we were able to do with funding from Rein in Sarcoma and the Altman Fund. And it's a small story, but a very, very important story. And I have only about 20 minutes or so. I can talk about it all day, but I have only 20 minutes. So I will skip some of the details. And if you have any questions, please stop me or ask me at the end. So our lab is also funded by pharma for our clinical trials and research, but this work is completely funded by Rein in Sarcoma. So the question is, why do we have to do research in rhabdomyosarcoma? It's very simple. There's an urgent and unmet medical need. We don't know about the resistance, recurrence, and metastasis that remain a major barrier to cure of these patients. And most importantly, there's an ethical reason. We have an obligation to develop safer and more effective therapies for our children with cancer. That is the purpose of our research. So the critical need for doing this kind of research is there are limitations. Standard chemotherapy has reached the therapeutic ceiling. We really can't advance the numbers anymore. There's significant treatment-related toxicities of these young patients and poor understanding of the resistant mechanism. We keep seeing resistance, but we do not know why this happens. So we, with the Altman funding, we met a number of times, we brainstormed, we talked about what are the ways in which we can advance the next generation of treatment for our patients. There are a number of ideas that came up targeting microenvironment. We tend to forget that the tumor exists in a microenvironment interacting with other cells of the body. That's another important way that we can attack this problem. And we need to come up with more ideas in the precision medicine approaches. We cannot treat all patients are the same. And most importantly, we all realize that immunotherapy is going to play a huge role in the coming years of advancing treatment for these patients. So this project kind of covers the whole spectrum of these new opportunities. That's why I really like this project. So how can we why do we think this is an important way to go about? The cells that we study, the models we generate came from patients. They give us mechanistic insight. We can study them, dissect them, and understand the biology. And most importantly, this will give us an opportunity for us to advance our drug screening platform. That's the precision medicine idea that I'm talking about, because in the old days, we used to take the tumor cells and look at a drug, whether the drug is working or not, but we forget the microenvironment. So we are moving towards recreating the patient's tumor microenvironment, asking the question whether a drug would be effective or not effective. And there is a huge difference in the activity of just studying the tumor cells and studying the tumor cells in their microenvironment. Obviously, in an ideal world, we can do this in the mouse models and so on and so forth. They are very difficult to do. They take a long time, and they don't. You really cannot have the practical ability to screen hundreds, even thousands of drugs to identify an effective target against the patient's cells. And there's an ethical advantage as well. This reduces the animal use while minimizing the importance, and also increase the importance of translational relevance. For all these reasons, we all realize setting up in vitro models is an important pathway to discover new treatments for our patients. So very briefly, the tumor cells do not exist. Tumor cells do not exist on their own. They are in microenvironment, especially the rhabdomyosarcoma cells are not isolated. They are in a dynamic niche containing immune cells, blood cells, fibroblasts, and so on. So they are in a complete ecosystem. And the tumor microenvironment, we are learning more and more. The tumor microenvironment plays a very important role in suppressing the immune system that is trying to attack these cancer cells. And therapeutic resistance also being mediated by these cells that we are learning very, very clearly. The drugs we give this patient kill those tumors in the lab when you treat the tumor cells with these drugs, but they do not work in the patient because these cells that surround and support these nursing cells actually prevent these drugs from working effectively. So they lead to therapeutic resistance. So this is our hypothesis. The fibroblasts play a role in the growth, survival, and treatment resistance of the rhabdomyosarcoma cells. They help the cells to proliferate and metastasize. We know that for sure. Now the primary sites, cells that surround the tumors in the primary sites, help to generate certain molecules and strength for these tumor cells to take off and go to a new site as a metastatic lesion. And they play a role in drug resistance, and they also play a role in immune evasion. All these things together provide the strength for the cancer cells to grow very, very aggressively and become resistant to our treatment pathways. So this cell line I'll talk about was established from a lung metastatic lesion of a patient, a very, very special patient who presented with PAX3-FOXO1 fusion protein. And after the ethics approval and informed consent from the family, we were able to receive the sample. We grew them in culture. Initially, we wanted to grow the tumor cells, but it so happened we were able to generate this very unique culture of these tumor-associated fibroblasts to study further and establish our model. So this is the pathway we took. First, we established and validated these fibroblasts as an important model. Then we looked at their functions, and they looked at what they are doing to support the growth of the tumor cells. And we also profiled on their own what they are making, what are the cytokines they are making, they are producing that can support the growth of the tumor cells. finally, ultimately, the most important thing is to find out how can we translate this knowledge into a treatment, an efficient treatment in the future. So first of all, we took the cells, and we have to make sure that these are not the cancer cells that we are growing. So we, as the figure shows, these cells do not have the PAX3-FOXO1 or MYC protein that confirm that these are not cancer cells from the metastatic lesion, and also they are positive for markers that are very typical of the stromal or fibroblast cells. So in our model system, we had a number of cells and cell lines. We use two cell lines that are rhabdomyosarcoma cells with the fusion protein, and we have used as a control a cell line for embryonal rhabdomyosarcoma without the fusion protein. And as a control, we also use a cell line that grows like this cell line, but came from a normal source. So first of all, we showed the cells promote the growth in a dose-dependent manner that the tumor cells. So what we did was we took the supernatant soup from these fibroblast cells that we established and added to the two fusion protein sarcoma cells and fusion negative sarcoma cells. And we showed that the supernatant, something coming out of these stroma cells, are able to stimulate the growth of these fusion protein positive cell lines. Then we also showed that these cells were able to stimulate over a period of time the growth of the fusion protein tumor cells. In other words, this might explain the rapid expansion of the size of the cell numbers in the metastatic lesions of the tumors. Then we asked the question: these cells that nurse and nurture these tumor cells, are they also help them to migrate, take off, and move along in their aim to metastasize? This is a very simple study experiment where you grow the sarcoma cells, make a scratch, and see how fast the cells migrate to close the scratch. So here you can see on the top, we incubated the scratch sarcoma cells with supernatant from a normal fibroblast and supernatant from our cell line, which showed a significant increase in the ability of the sarcoma cells to migrate and close the gap when they have access to the supernatant from the cell line that we generated. So, but it did not work so well in the fusion protein negative tumor cells. So whatever the activity is coming out seem to have some preference or some specificity to the fusion positive cell lines. We were able to reproduce that with a number of other fusion protein cell lines as well as primary tumor samples. So then the question is, why do these children fail to mount a very strong immune response? Their immune system is good, they're functioning perfectly, but somehow the tumor was able to block the activity of the immune system. So one of the things that everybody's looking at is called the checkpoint inhibitors. These are molecules that come out of the tumor, and they bind to certain T cells and tell the T cell not to react against them. So we actually showed when you have the supernatant, the tumor cells were able to increase these checkpoint molecules to inhibit the T cells. So that might be one of the mechanisms. We do not know if this is the only mechanism, but it seems like it's one of the mechanisms by which the immune system fails to attack these tumors. This is important because we do have checkpoint inhibitors now. So there are a range of pharmaceutical companies making molecules to block this pathway. So one of the things that we found, I'll talk about this later, we found a cytokine called IL-6 that comes out of these fibroblast cells. They can activate a pathway called STAT3 pathway. Then that can increase the synthesis of the checkpoint molecules. So we want to know the mechanisms. Like I mentioned at the very beginning, we want to attack all three things. We want to have new drugs. We also want to know the biology. We want to know the mechanisms. So this seems to be a possible pathway by which that these tumor cells were able to increase the checkpoint inhibitors and evade the immune activity. And then we also screened a whole range of chemotherapeutic agents, and we showed in some of them the stroma cells were able to block or enable the tumor cells to become resistant to certain molecules, not to all chemotherapies, but to certain chemotherapies. This resistance seemed to help. Here, for example, I show cyclophosphamide. You need more chemotherapeutic agent to kill the same number of tumor cells if they have access to the supernatant from the fibroblasts. So they resist it. This is also important because now we have a way in which we can actually screen some of the chemotherapies in vitro to give the oncologist an idea which chemotherapy may work or may not work in that particular patient. Again, going back to a personalized treatment approach. This is, we screened hundreds of cytokines. We want to know exactly what these cells are making. As you can see, some unique cytokines are increased quantitatively and qualitatively, if everything else being normal. Here, we took from the same number of normal fibroblasts and our sarcoma-derived fibroblasts, and looked at the difference of the cytokines each one of these are making. So now the question is, how do we connect the cytokine data to what we are seeing as a function of these molecules? So we showed that some of these cytokines we found to be increased also have a function of upregulating the tumor functions, migration, metastasis, chemotherapy, angiogenesis, the generation of blood vessels to the tumor that will help the tumor to grow, and so on and so forth. And the opposite is also true. The supernatant from the tumor also blocked a number of very important immune factors. I agree these are all in vitro studies, but it gives us an idea how aggressive these tumor-associated fibroblasts in their quest to support the tumor to grow and survive and metastasize. These are all the molecules that are downregulated in the molecule that also play a role in the immune system. And finally, we wanted to see what is the mechanism, and we showed that there are certain pro-survival, pro-growth pathways are activated when these two cells come together as a pair. When they are near each other or they have access to the molecules the other one is making, the very specific pathways are activated, the STAT3 pathway and the MAPK pathway. This is also very important because there are a whole slew of new drugs coming up through the pipeline that are safe and can be effective against targeting this pathway. In the future protocols, we can consider including these very unique targeted therapeutic agents in the treatment of these children. So we wanted to focus on, okay, now we have the idea, now we have the data, but we have to move along finding new treatments. So one of the things we found was the pathway called SDF1-CXCR4. Is a signaling is known driver of fibrosis in other cancers, lung and breast cancer. We know when the microenvironment makes these cytokines, the tumors grow really, really aggressively. Then we show in our data, similar cytokines are activated. So putting one and one together, we wanted to explore further, can we get a drug that can target this pathway that we can show that it works? So we found a drug called NM92 922. It's been tested is FDA approved for certain indications, and it's been approved for other cancers. So it seems like a safe, even though there's no data available on this drug at that time when we are doing on pediatric cancers, especially sarcomas. And so multiple factors regulate the activation of this pathway, and there is a therapeutic potential because it drives, we know from breast cancer and lung cancer models that this is actually a molecule that drives metastasis. And so we want to see if that's also true for sarcomas. So we showed that these cytokines are upregulated when we treated these supernatants together, and the NM922 effectively targets the CXCR4 in these fibroblasts. So now we have a drug that attacks the fibroblast, the helper, instead of going after the tumor directly. And finally, we showed that the fibroblasts are twofold more sensitive to this drug compared to normal. So that's important because we don't want to have non-specific toxicity against normal cells, normal fibroblasts in the body when we are aiming to treat these patients with such a molecule. So this is the model. This is what happens normally. When we block it, we may be able to block the IL-6, MCP1, and VEGF-A regulated pathways. So what are the therapeutic implications? The stromal targeting supports microenvironment, dual targeting potential, and combination strategy. So we can take the drugs that seem to work directly attacking the tumor, then we can combine with drugs that blocks the supporter of these cancer cells to increase the efficacy of our treatment. So previous reports suggested, they are not found in RMS, primary tumors. Primary tumor discovered having very little of the stroma. In vitro studies show limited interaction with normal fibroblasts. But our model created that in metastatic lesions, which is very important, that these cells play a role. And we also were able to find some of the biological and critical pathways of these molecules. There are some limitations. It's a single patient-derived model. We need to increase this further to confirm it. And condition-mediated approach captures soluble factors only. There may be factors that do not get released by these cells, so we are in the process of studying them. We misses direct cell contact when we are looking at supernatant. Multi-patient, what we are doing now, we are trying to get more patient sample, see if we can reproduce that. We are also sharing these cells with other investigators across the globe to help their studies. That's one of the things that the family wanted us to do. So we can increase the research that's going on using our model. And we are also in the process of validating that in mouse models. We have mouse models for the sarcoma cells, and we are generating data on that. So to summarize, the metastatic cells are reprogrammed to secrete pro-tumorigenic factors. These cells drive proliferation, migration, immune blocking, and drug resistance. And targeting the CXCR4 axis, especially in combination with the other molecules, that can increase the efficacy of the treatments. So this approach will enhance the safety and activity of new therapeutic approaches that are coming up. And as we know, CAR T cells in my lab, we have developed a cancer vaccine to target these fusion proteins. So one of my students is studying how can we increase the activity against immune therapies like CAR Ts or cancer vaccines by targeting these molecules. And these fibroblasts gives us a fabulous model to analyze and validate those molecules. For example, one of my students using CRISPR technology to silence individual genes and to see how we can identify the genes that help our future immunotherapeutic approaches. So this is my team, the students who work with me on this project, and these are the funding sources for our project. And thank you, and I'll take any questions if you have.
So questions for Dr. Narendran? Maybe I'll start with one. So I guess I'm wondering whether you think that there are just one type of cancer-associated fibroblasts in these metastatic niches, or more than one type, because in the literature, people have talked about different types, some heterogeneity within them, myofibroblasts versus antigen presenting or inflammatory fibroblasts. Looking at the primary metastatic tumors, do you see evidence for that heterogeneity within the fibroblasts?
Yes, absolutely fabulous question. So I tell my students that this is a functional definition. There are no such thing as a stromal cell. It's the functional definition. I look at them from the point of view of what they are doing to the cancer cells rather than what they are. So that's exactly what you're alluding to. So if you start the stromal cell culture, you will have fibroblasts, you have macrophages, you have osteoclasts, osteoblasts, and so on and so forth. So the whole slew of cells. I feel that they all have a role to play. They kind of synergize with each other in the body, and we were able to pick the cell line that actually supporting the cancer cells when they are able to grow and become a culture. In other words, we probably created a cell line that covers or carries the function of the multiple cell lines in vitro. So it's kind of artificial. It may not even exist in the patient, but somehow it picked up the function or selected out for the functions these cells provide. So yes, there are multiple cells. So the proof is going to come when we started doing animal studies. Then we can test your hypothesis, whether is it a one single stromal cell pathway, or is it the whole family of molecule of cells that support these tumor cells. Yeah, that's a very important question.
I have another question here.
Yeah, Andrea is one of the medical oncologists. I was wondering, what did you find the embryonal randomized sarcoma cell lines and their stroma, if there's some similarities or it's a completely different mechanism?
Yeah, so I admit we didn't screen a huge number of cell lines, very small number of embryonal and cell lines. Somehow there seemed to be a quantitative difference. We do know why that is. The best way to do is generate a similar cell line from embryonic sarcoma and see if the reverse is true, or my hunch, I have no proof. My hunch is the fusion protein generates some kind of an activation pathway that is targeted by the molecules that come out of the stroma cell line. So it's specific because of the aggressive molecules generated as a result of the fusion protein. So, for example, we took some neuroblastoma cells, pediatric leukemia cells. My lab is full of these pediatric cancer cell lines. We found that there seems to be some kind of a bias in supporting the fusion PAX3 FOXO1 fusion protein positive cell line, and it's not 100%, but there seems to be a bias by these factors. But that's that I think that's kind of explained by the fact there's not one single factor. Is the critical mix of molecules that seem to support these patients. As you know, we are learning more and more about how simple things like starvation can affect how the immune system reacts against a tumor. So there are many different variables that is playing a role in this. So that'll be an interesting question to pursue, whether this is a fusion protein specific issue, or is it true for all of the cancers?
Other questions in the room? Did you get me one more? All right. Well, thanks so much, Dr. Narendran.
Thank
So our
you.
next speaker is Dr. Nia Friede, a resident pathologist at the University of Wisconsin-Madison. Nia is a Jan Modlin Sarcoma Scholar Program alum. She collaborated with other scholars on the study she'll talk about today, which led to it being published in the Journal of Cancer Education last year. Today, she'll present on assessing sarcoma awareness among the general population in Minnesota, a cross-sectional survey study from the Minnesota State Fair in 2015 and 2022. So please welcome Dr. Friede to the stage.
Give us 1 second just to get her. Here. This one?
Should be the only one on them.
Yeah, that looks great.
Beautiful.
Perfect.
Looks great. Is that it?
Yeah.
So let me share really quick. So- Sorry, they told me to bring one on a flash drive, and I had to find a flash drive, which I have not used in maybe 5 years. So my name is Nia. I'm a second-year resident physician at University of Wisconsin-Madison, and I was a Rein sarcoma Jan Modlin Scholar in 2022. There we go. So as with everyone else here, this work was supported by Rein Sarcoma and then the Driven to Discover program at the University of Minnesota. It's not quite right on. Sorry.
No, go ahead.
So sorry.
That's why I wanted you to set it up. Thank you.
It's always so tricky.
So I just want to say first and foremost that this was a very collaborative effort. I am just the face of this. I was lucky enough to be close by to present this information that we published in 2024 in the Journal of Cancer Education. So we're really a diverse group, and that's one of the big things that I really enjoy about Rein sarcoma and my time as being a sarcoma scholar is that I got to work with people that really led my practice moving forward. As a pathologist, sometimes we are kind of back in the corner. So working with radiation oncology, pediatrics, internal medicine was really a great thing to do. Out of this list, 8 of us, okay. Were sarcoma scholars. So this was really the brainchild of Dr. Amber Resclaff, who is a radiation oncologist now at Abbott and Allina here. So she started this in 2016 when she was a Jan Modlin scholar, and I'm sure most people here are aware of it, but those who may not be online, this is an annual scholarship that is awarded by Rein sarcoma with the goal of educating the medical community and the public about sarcoma.
And then during your 1 year appointment, the sarcoma scholars would attend various events and then also participate in research projects. So our project was done at the Driven to Discover Research Facility at the Minnesota State Fair. This is a tent at the State Fair for anyone who goes to the State Fair, which is everyone. You should always go to the Driven to Discover tent because the people are there trying to get more people to do their research and fill out surveys, and it's really, really helpful. So drag your families, make them go to the tent, take 20 minutes and sorry. Oh, well, I mean, I was there, so. Okay. The building. To go there and fill out those surveys, because it's really helpful to everyone. We did have Rein and Sarcoma scholars there on other years, but these were just the 2 years that we had access to our full data sets. So the project inclusion, our inclusion criteria, we required the participants to be aged 18 or older. They needed to be English speaking. We only had the survey written in English. They had to be current residents of the state of Minnesota. That being said, we didn't check IDs. We just asked if they are current residents. And then the participants had to be able to fill out the electronic questionnaire on their own. And obviously, Rein and Sarcoma members and the volunteers were excluded. So the people who met the inclusion criteria and wanted to participate were given an iPad that had a 3-part survey on it. The first page, of course, was an electronic consent with our IRB requirements. And then from there, they filled out the 3-part survey, which was all multiple choice questions. On average, the survey lasted about 10 to 15 minutes. And we had 2 to 3 people there at a time manning the station. So it was broken up into these 3 parts. The first part of the survey was our attempt to find out what people would do if they or a loved one developed a lump or a bump, which is a common presentation of some types of sarcoma. We asked how quickly they would try to see a provider and then who their first line provider that they would try to see. So part 2 of the survey was our attempt to quantify and assess sarcoma awareness. Participants were asked to answer 10 multiple choice questions listed here, and we'll go over these in more detail. All the questions were related to sarcoma. The questions covered the definition of sarcoma, the incidence, the subtypes, the etiology, common age groups affected, what body sites are affected, tissue types, common symptoms, and more. Part 3 was demographic information. We asked about age, gender, level of education, previous employment, healthcare, and their zip code. The demographic information was important to us because the goal of the research project was really twofold. One, to get people talking about sarcoma. We wanted to educate. And so after they filled the survey, a lot of times we would have questions and we would get to talk to people about red flag symptoms. And then 2 was to assess the understanding of sarcoma in the state of Minnesota itself and where we can provide education. So to comment on that, we needed to have an idea of who was actually filling out the survey and who was coming into the research building and filling out this survey. So we actually used the zip codes provided and subclassified the participants into one of 4 categories, which is defined by Minnesota State Demographic Center, into either rural, town, rural mix, urban, town, rural, and then entirely urban. So that was the breakdown of the survey that we stood and handed out and talked to everyone about. So this was the breakdown of our survey information, and kind of what we were hoping to accomplish. So this is what we got. In 2015, we had 1,491 participants who filled out the survey. In 2022, we had 633, which gave us a total of 2,124. There are multiple reasons for why there's such a drastic change in the number of people that filled out the survey. Primarily is about how many days we were able to get people to man that research table, and how many volunteers we had every day. And for reference, the Labor Day attendance in 2015 was 178,000 and 150,000 in 2022. We're still very happy with these numbers overall. And as you can imagine, sometimes it's hard to get people to fill out survey after survey in that building. So we compiled the data, and actually the data from 2015 to 22 was very comparable. So we could actually get to put it together. The statistical analysis was performed by Dr. Alexander Nelson. He's over at BIDMC in internal medicine. And for part one, he looked at the association of timing of evaluation between a painful versus a non-painful mass. And then he used a chi-square test to assess the provider choice. For part two, we looked at sarcoma awareness by how many questions were correct. And then finally, he did linear regression analysis to look and see if sarcoma awareness was correlated with any demographic parameters. So part one, timing of presentation. So the first thing we asked all 2,124 participants was how soon evaluation would be sought for a non-painful mass. So out of that group, 26% indicated that they would make an appointment as soon as possible. In contrast, a painful mass, 50% of participants indicated that they would make an appointment as soon as possible. This is very relevant to something like sarcoma, which presents often as like a non-painful mass. So you see half as many people would go in sooner. 5% of participants would never seek evaluation for a non-painful mass, whereas 1% would seek full evaluation for a painful mass. Overall, we see that individuals were more likely to see medical attention sooner for a painful mass compared to a non-painful mass. The second part of one is provider choice. So both non-painful and painful masses, the majority, so 77% in each category, indicated they'd make an appointment with a family medicine physician first. There was no difference in the provider choice between painful or non-painful. And the next most common provider listed were advanced practice providers at 10% and 9% respectively, and internists then at 7%. This is really important, I think, for Rein and sarcoma because it kind of can tailor in the medical community who we should focus our education literature towards. And I remember when I was a sarcoma scholar, we did a lot in family practice journals and pediatric journals, and because they're really our first line clinicians. We also always had listed as none of these options for every multiple choice because we didn't want our participants to have to feel like they had to choose an option. So part two was sarcoma awareness. And this was a list of the 10 questions that we had. So again, all of the survey questions had an option of none. And so for the first question listed here was just, what is sarcoma? And the correct answer is a type of cancer. And 67% of participants got this question correct, which as a pathologist, this makes me very happy. Our next question was, what age groups are affected by sarcoma? The correct response was that all age groups are affected, and this had a 50% correct response rate. Our goal of this question was to communicate that there's not a specific age range for sarcoma. Question number 3 was what causes sarcoma? Since there are many types of sarcoma and many driver mutations, which we had some exciting lectures already on, the correct answer is really none of the above, which got 32% correct. Question number 4 had a very similar goal to question number 2. We wanted people to know that sarcomas can really present in many locations, and that sarcomas are tumors of many tissues, that a lump of the bump of the neck or the arm, or even abdominal swelling could be a primary presentation of a sarcoma. And this question got 49% correct. Question number 5, again, had the same goal of teaching our participants that sarcoma does not come from one place, but can come from various tissue types, and 34% got this correct. Question number 6 had the worst correct response rate with 14%. And this question was, how many subtypes of sarcoma are there? And I honestly don't even know the answer to this question because the answer is a lot. For question 7, we wanted to communicate that sarcoma it can present in many different ways. Like I mentioned before, it can present as swelling, a pain at night, or a lump and bump, or even just overlying skin changes. And this correct response rate was 48%. Question 8 was about treatment, and this was 46% correct. Question 9 was incidence, and 16% of participants got this correct. Sarcoma diagnoses in the United States are between 10 and 30,000 new diagnoses when we were asking the survey. In comparison with colorectal cancer, which has about 150 new diagnoses per year, and breast cancer, which has 300,000 new diagnoses per year. And then our final question was, what would be beneficial to patient outcomes? Basically, the things we would like people to be doing going forward. We would like better public education and physician education about the signs and symptoms of sarcoma. We would like patients to make appointments with clinicians sooner rather than later when concerning signs come up. And finally, we'd like our primary care colleagues to be cautious when excising non-biopsied soft tissue masses. And 47% of participants got this question correct. Overall, 24% of participants scored 70% or greater, and less than 1% of participants correctly answered all 10 questions. On the other hand, 16% had no correct answers. Part 3 of the survey was the self-reported demographic of our survey takers. When comparing this data with the scores of Part 2, we found among self-reported gender identity that males were estimated to perform 1.015 points worse on survey completion compared to the female participants. In comparison to the reference age group of 18 to 27, older age groups performed better until the age of over 68, in which there was less effect noted. In comparison to individuals with some high school education or GED, all other levels of education performed better. Individuals with a graduate or professional degree had the largest estimate in increased score at 2.5 points. And then participants without current or previous healthcare employment performed worse at an estimated decrease of 1.79. There was no difference in overall score when stratified by county categorization, although the majority of participants originated from entirely urban counties. And then here's the picture of the geographic distribution of survey participants based on the zip code they provided. We can see that the majority of attendees were from the Minneapolis-St. Paul area and surrounding suburbs. The next highest level were more outer-lying suburbs, as well as the county that holds Duluth, Minnesota. And this data corresponds to population densities of these areas. So, results from part 1 of the survey demonstrated that pain is a driving factor for patients to seek evaluation, and that family practice providers are the most likely first point of contact. This underscores the importance of sarcoma knowledge, particularly among primary care providers. Results from part 2 of the survey demonstrated a deficit in regard to sarcoma awareness in the general population, with scores trending upwards with a higher level of education and healthcare employment background. This highlights the importance of healthcare providers focusing outreach efforts on those with lower levels of education and no healthcare experience. Although overall scores were better among individuals with a healthcare background, participants in that category still scored an average of 55% correct, which highlights room for improvement in this cohort as well. Education on sarcomas is scarce in medical education and kind of likely contributes to these scores. In our 2015 and 2022 cohorts, well over half participants identified sarcoma as a type of cancer, although under half were able to identify symptoms. This highlights a key area of concern, as early diagnosis is critical for effective treatment, and the majority of the general public in Minnesota is unaware of red flags of sarcoma. So sarcomas are commonly misdiagnosed, and treatment delays negatively impact patient outcomes. The purpose of this study, let's go back one, was to explore patient threshold for and timeline to a medical evaluation, to identify providers most likely to be contacted first, and to assess general sarcoma knowledge in Minnesota's general population. We found that even with a robust nonprofit organization like Rein and Sarcoma in the state of Minnesota, that sarcoma is still poorly understood. We found that even those with advanced degrees in healthcare experience had lower than expected sarcoma awareness, and we believe that these results should help bolster Rein and Sarcoma's goal of increasing sarcoma awareness in the general public. We also hope that our time in the Driven to Discover building provided education for the participants we interacted with. So that was it for me.
So, questions for Nia?
Thanks, and sorry, I didn't mean to correct you. I just spent a long time getting that building built, so I didn't want us downgraded. But I realized in 2015, and you might have actually been in a tent out front. I'm curious if you saw any difference, maybe I missed it, between the 2015 and 2022 respondents?
No, that's why we pooled it, honestly. So this was Amber's baby, and she did it in 2015, and they did it again in 2016, and I think 2018, and then COVID hit, and obviously we took a bump back. And our biggest numbers were 2015 and 2022, and we thought when we ran the numbers, we were expecting to see that. We were hoping to see a change in the data, but I think even on the primary provider, it was exact same percent of people, 77% were going to go to their family practice doc. So I was very surprised by the fact that there was no change in the data that we saw.
I mean, honestly, I'm not, because had lots of surveys there and over years, and what you find is, you're basically sampling cross-sectionally, like public opinion and knowledge, and Rein and sarcoma is great, but the state is very large, and I think that without a coordinated statewide campaign to make people more aware of sarcomas, I wouldn't expect that people are going to get more aware in the space of 7 years, especially with big things happening like COVID in between.
Right.
Yeah, but I'm also just absolutely tickled that you got so much usable, actionable data out of little old survey at the fair. So
Well,
thank you.
yeah, no, I went with them in 2022, and I said, we have all this data. Why aren't we doing anything with it? It just got sat on for whatever that was, 8 years. And then I basically had to poke at people smarter than me to run the numbers and get this out there. Because, what's the point of doing this without making it kind of actionable?
I personally think that funders and research for it is that the word sarcoma is very scary. I mean, and it's legitimately scary because they're obviously hard to treat. But it brings to mind like sarcophagus, or it is a scary word. I think that we as a community should spend more time actually getting into the public's consciousness. I don't recall, did you even ask whether they realize whether sarcoma is a cancer?
Yeah, that was our first question. That was the one that I was happy about because it was 76%. I'm sure there's some sort of bias for being in that space. But like especially after COVID, like infectious was one of the options, things like that. So that was our first question.
I mean, not number of people.
Mhm.
Thank you.
Thank you.
I was just curious if your team had any plans to try to do a statewide push for education to be more accommodative family practice setting.
Yeah, so actually when I was with Rein and Sarcoma, that was one of our big goals. So we had been putting in not advertisements, but in our like blurbs and like family practice and pediatric magazines, we would put like a little Rein and Sarcoma advertisement, quote unquote, but it would have like an important fact, like, did you know that the non-painful mass the size of the golf ball might be the first presentation of a sarcoma, and things like that. So we were already, before we published this paper, when I was there, we were tailoring towards that effort. And we were just doing not industry-wide, but physician-wide journals at that point. So I know that I was located in Duluth at that time. So I did specific education at Duluth because Duluth is a big city, but the patient population that we see up there is primarily rural, coming from northern Wisconsin, North Dakota, and up in the North Woods there. So I did a lot of education with the
radiation oncology group there. And I think this information and what we were doing, we were we were doing the education in the radiation oncology area, and then we realized like those people know what sarcoma is, or they know what cancer is. They're a little bit more literate than people who are just going to the pediatrician or their family practice. So we started putting more information in those areas. So that's kind of how we changed our tailoring of spreading information at that point.
Thank you.
Yeah, of course. Oh, I don't want to check that. Thank you.
All right, so last minute substitution, I'm going to be presenting the next topic, which is combination mirdametinib plus fernostat for malignant peripheral nerve sheath tumors. Did I just go forward?
It's like 10 minutes ahead of schedule, so just.
We can have a break after. Let's be-
Sure.
We're good. Does that look good? All right, thanks so much, Abby. There we go. All right, so these are my disclosures that have to do with the companies that I work with, but none of the work that I'll talk about today is really related to the business of these companies. I will, though, describe a clinical trial that we have open where we're getting free drug from SpringWorks Therapeutics. So today I'm going to talk about neurofibromatosis type 1 syndrome, which is truly our sarcoma predisposition syndrome, among other types of cancer, the peripheral nerve sheath tumors that develop in NF1, how we're leveraging synthetic lethality and synthetic drug sensitivity to try to develop new treatments for these peripheral nerve sheath tumors, and then finish with the window of opportunity trial we have open that features combined MEK and HDAC inhibition. So what is neurofibromatosis type 1? This is a common autosomal dominant cancer predisposition syndrome. It affects about 1 in 3,000 live births. Although it is an inherited disorder, 50% of cases are de novo, meaning the affected person is the first in their family to have it. So neither parent has it, but the child does. And so what happens is that this gene can become mutated in the germline, in a germ cell, mom or dad. So I've met many families who've never heard of the disease, and out of the blue, they have a child affected by NF1. All patients with NF1 syndrome have cafe au lait spots, Lisch nodules, which are benign hamartomas on the iris, and dermal neurofibromas. And those features help pediatricians diagnose this disease in children. But it is a complex syndrome, and so there are other manifestations of the disease that may happen, such as skeletal abnormalities like scoliosis, endocrine defects, and problems with learning and memory or attention deficit disorder. These patients are also prone to benign and malignant tumors. The most common cause of disease-associated mortality is malignant peripheral nerve sheath tumor, or MPNST, a soft tissue sarcoma. Now, the cause of NF1 syndrome is inheriting one loss of function mutation in one copy of the NF1 gene that encodes neurofibromin, an important RAS GTPase activating protein, or GAP. It's a negative regulator of RAS, and so you can imagine when it's lost from cells, you get increased RAS signaling, and that can drive neoplastic cell growth. It's worth remembering that NF1 is also an important tumor suppressor gene in sporadic cancer, not just in NF1 patients. So colorectal cancer, lung cancer, leukemia, glioblastoma, a percentage of all of those have mutations in NF1, even in non-NF1 patients. So some traits in NF1 are associated with the fact that the patient has inherited one loss of function copy of the gene, such as the cognitive defects. Those are thought to be due to haploinsufficiency, the fact that there's just one functional copy of the gene and not two. But all these other effects of the disease, these benign and malignant tumors, these different forms of cancer, they're due to somatic loss of the wild type allele in a rare cell, so that you end up with a cell that's got no functional copies of the NF1 gene. Okay. This disease is complicated, and you need special training and special clinics to help follow patients with NF1. And what they found out is that the complications are dependent on age. So some of the defects you see in NF1 syndrome are congenital. The patients are born with them, like the plexiform neurofibroma or juvenile myelomonocytic leukemia. Some develop in early childhood, like optic pathway glioma, and some develop in adulthood, like the malignant peripheral nerve sheath tumors. But it is definitely a cancer predisposition syndrome. So almost 60% lifetime risk of cancer in NF1 patients. The 5-year survival rate for cancer is worse in NF1 than in the general population, and they're 10 times more likely to develop any cancer. 40% of patients will develop tumors other than the neurofibromas that give the disease its name. And so these are some of the tumor types that occur with increased frequency or prevalence in NF1 patients. So it includes sarcomas like MPNST, but also other sarcomas too. So it's like osteosarcoma and others, gastrointestinal stromal tumors. So actually, this disease unifies a bunch of the different translational working groups that we have in the Masonic Cancer Center, in the sense that these patients are predisposed to brain tumors, predisposed to sarcoma, predisposed to leukemia, and breast cancer. Now, just to elaborate a little bit more on the peripheral nerve sheath tumors that develop in these patients, they all develop from Schwann cell lineage. Within the peripheral nervous system, Schwann cells are the predominant glial cell type that are famous for myelinating the axons of peripheral nerve cells. Schwann cells are a derivative of the neural crest in development, and they give rise to Schwann cell precursors that can differentiate to myelinating Schwann cells or non-myelinating Schwann cells. And so their role is in supporting peripheral nerve cells and assisting in repair if the nerve is damaged. These malignant peripheral nerve sheath tumors develop in NF1 patients along a specific histopathological pathway. So as I mentioned, the patient with NF1 has inherited one mutant loss of function copy of NF1. Whoops. And in a rare Schwann cell or Schwann cell precursor, you can have loss of the wild type allele, and that, along with other changes like inflammation or wounding, are thought to lead to the plexiform neurofibroma. That can progress to an atypical premalignant stage called atypical plexiform neurofibroma via loss of cyclin-dependent kinase 2A, cyclin-dependent kinase 2B genes, important tumor suppressors. That in turn can progress to MPNST, and in 80% of cases, that's associated with loss of the polycomb repressor complex 2, PRC2. And that happens by biallelic inactivation of EED or SUS12 that are critical components of the PRC2. Now, this transition to MPNST is also accompanied by a bunch of other changes. So you have aneuploidy, you have gene copy number gains and gene copy number losses occurring as well. So it's not as simple as just the loss of these tumor suppressors. Other things are happening too. Nevertheless, RAS-activated signaling pathways are likely to be useful targets for NF1-related tumors. I mentioned that the product of the NF1 gene is called neurofibromin, and it is a RAS GTPase activating protein. So RAS is an important signaling molecule downstream of receptor tyrosine kinases, for example, in cells. And when RAS is bound to GTP, it's active in its signals. Gaps like neurofibromin interact with RAS GTP and greatly increase its intrinsic GTPase activity. So when neurofibromin interacts with RAS GTP, RAS will become activated, cleave off this third phosphate, and be converted to RAS GDP, the inactive form. Now it can become activated again by guanine nucleotide exchange factors, but this is basically the off switch for RAS signaling. So when it's lost from a cell, RAS GTP accumulates to too high a level and it stays on too long after growth factor signaling, for example, through receptor tyrosine kinases. And so as a consequence, you get increased and prolonged signaling of RAS activated pathways like the RAS MAP kinase pathway, the PI3 kinase mTOR pathway, and other pathways. And that's what drives neoplastic growth, it's believed. And because of that, targeting those RAS activated pathways would make sense therapeutically. And in fact, the MEK inhibitor selumetinib is FDA approved for plexiform neurofibromas that are clinically problematic. So where they're growing too big, pressing on a vital structure. And this is a figure from a paper published in New England Journal showing that an MRI of a plexiform neurofibroma, and they can get very, very large, you can shrink it with treatment with selumetinib. And this led to the approval of this drug. So in most cases, you can get 20 or 30% reduction in size of the plexiform neurofibroma with selumetinib. Chris Mortel, who until recently was running the NF clinic here at the University of Minnesota, he collaborated with SpringWorks to show that another MEK inhibitor called mirdametinib might work even better. And this is now FDA approved for adults and children with clinically problematic plexiform neurofibromas. And this waterfall plot just shows the reduction in tumor size after treatment with selumetinib, which a patient has to take every day for this to happen. And if they stop taking the selumetinib, the plexiform neurofibromas often start growing again. Now, what about the MPNSTs? Well, right now, the only real way to cure one is if you can get surgery with clean margins, but that's often not possible to achieve because of where the MPNST is growing. Taking it out or all of it out could be too morbid for the patient, or you could imagine it growing on a nerve that innervates the diaphragm, and so you literally can't take it out or the patient wouldn't be able to breathe. So instead, radiation and adriamycin-based chemo are used, but local recurrence is common with the eventual development of metastatic disease so that 50% of patients are dead by 4 years. Unfortunately, there seems to be little response to single-agent MEK inhibition for MPNSTs. And the field has tried a number of clinical trials for MPNST of drugs that target tyrosine kinases like EGFR, VEGFR, or drug combinations, but so far nothing has really worked. Now, the genomics of MPNST don't suggest obviously frequently activated oncogene targets that we could use for therapy. So instead, as I was emphasizing, these tumors develop as the stepwise loss of tumor suppressor genes. So the loss of these tumor suppressor genes are removing barriers to transformation. First, loss of NF1 that causes neurofibroma, then loss of CDKN2A and B that causes atypical premalignant neurofibroma, and then loss of the PRC2, along with other changes that leads to MPNST. So what we've done in the lab is try to figure out what drug sensitivities result from the loss of these tumor suppressor proteins. And I've already told you that MEK inhibition is useful at the neurofibroma stage to shrink tumors and prevent them from getting bigger. Work that we've done in the lab shows that thymidine biosynthesis or MDM2 inhibition might be useful for the atypical neurofibroma. I won't talk about that today. I will talk about how HDAC inhibition seems to be useful for treating PRC2 deficient MPNSTs We've also explored the cell surface of MPNST to try to find additional targets for therapy with the idea of potentially developing CAR T cell therapy in collaboration with other labs. So in our lab, we're really thinking about these peripheral nerve sheath tumors in two ways. What are the intrinsic vulnerabilities that they have because of loss of these tumor suppressors? And what are the extrinsic vulnerabilities that we can exploit on outside of the cell? So we've performed drug screens in CRISPR-edited immortalized human Schwann cells, as well as whole genome synthetic lethality screens with CRISPR libraries, and developed human iPSC and mouse primary Schwann cell models for testing these ideas. So this is a schematic of the whole neurofibromin protein, and you can see in the middle the gap-related domain. So the protein has a bunch of other functions associated with interaction with these other proteins. When NF1 is mutated in a cancer cell, usually there's no neurofibromin protein expression at all. It's completely gone. And so all these interactions aren't happening. And so it's got other functions that we can't predict easily what sensitivities might result from loss of neurofibromin in a cell. And also synthetic lethal effects are likely to be context dependent. So basically we're asking, when a tumor cell doesn't have neurofibromin and all these things aren't happening, are there vulnerabilities that cell has that a normal wild type cell wouldn't have? And so to explore that, Kyle Williams in the lab, who's now transitioned to independence in the Department of Pediatrics, he took immortalized human Schwann cells and made NF1 knockout clones, isogenic clones from those cells using CRISPR so that we could compare the cells that are identical except for loss of NF1. And he did that by targeting exon 10 with CRISPR, and he was able to isolate clones that didn't express neurofibromin anymore, as shown in this Western blot, as well as sister clones that continued to express neurofibromin protein. As expected, the knockout clones have increased basal levels of RAS-GTP, consistent with its known biochemical activity as a RAS-GTPase activating protein, so that when neurofibromin is gone, RAS-GTP levels rise. These knockout Schwann cells were also partially transformed in the sense that they could form colonies in soft agar and also grow as tumors when implanted into nude mice. So Kyle did a large drug or small molecule screen with these cells, comparing the wild type cells and the isogenic NF1 deficient cells, looking for drugs that would kill these mutant cells and not the wild type cells. And working with Gunda Georg and John Hawkinson in the Institute for Therapeutics Discovery and Development, screened over 11,000 drugs, including all FDA-approved drugs, looking for drugs that would have that trait. And, we chose drugs that were approved or in clinical development and been tested in people for many of these drugs because, in part, we were thinking this is a drug repurposing screen. There might be drugs already out there that are useful for treating these patients. We just don't know about it. And the translation of these findings is going to be a lot faster if you can use a drug that's already known. We already know safe levels of that drug to use in people. So he found some drugs after doing that kill NF1 mutant cells more readily than isogenic controls. He found about 20-something drugs that had that trait of the 11,000, and they fell into certain activity classes. For example, a whole cluster of drugs that increase intracellular calcium as an effect of their mechanism of action showed greater activity against NF1 mutant cells than wild type cells. A drug that does the opposite actually had the opposite effect, where it killed wild type cells better than NF1 mutants. So we were pretty confident about this result. But there were other activity classes too, like drugs that inhibit NF-kappa B. So we tested these drugs in a genetically engineered mouse model of rapidly developing 100% penetrant plexiform-like neurofibromas in mice where we induce genetic deletion of NF1 as well as a second tumor suppressor gene called PTEM. The justification for doing this is that many human plexiform neurofibromas downregulate expression of PTEN. So we felt like this was an okay model to use. Also, because the mice develop tumors really fast, we would get an answer for whether the drugs would work quickly using this model. We also tested the drugs versus human MPNST T cell line xenografts. All the drugs that we picked were partially effective in both the genetically engineered mouse model and human MPNST xenografts. This shows percent survival versus days after the initiation of treatment with these drugs. And untreated animals had to be euthanized very quickly in this genetically engineered mouse model, but the drugs all prolong survival to some extent. The same was true of mice that were engrafted subcutaneously with human MPNST's cells. And then Kyle went on to show that if he started to combine a MEK inhibitor with some of these new drugs, he could get these plexiform neurofibroma model mice to survive even longer. So this is selumetinib plus a drug in development called rigosertib, or selumetinib plus a heart failure drug called digoxin, which is already used in patients. So this data for us suggests that these drugs might make the MEK inhibitors work better for plexiform neurofibromas and allow us to use lower levels of the MEK inhibitor, for example, or the patients to take drug holidays. But we also think they could be used to delay or prevent progression, as we found they're also active versus MPNST. Now, the next thing I want to talk about is what happens when the MPNSTs lose the polychrome repressor complex 2. So this is a schematic of the PRC2. And SUS12 and EED are parts of the core complex of the PRC2. It's important in maintaining cellular identity. And its job is as an epigenetic writer. So it modifies histones. It causes the mono, di, or trimethylation of histone H3 at amino acid position 27. So a very specific methylation mark. And that is associated with repression of gene expression. So when the PRC2 is lost during tumor development, you get inappropriate gene expression. And Minu Budi in the lab published a paper earlier showing that about 1,200 genes get abnormally expressed as a consequence of PRC2 loss. And some of those genes are driving cancer progression. As part of that work, we modeled loss of the PRC2 in these NF1 knockout human Schwann cells. And you can see in this Western blot that we were able to knock out SUS12, and that results in complete loss of histone H3 lysine 27 trimethylation as expected. And so we were able to use these cells to do additional drug sensitivity comparisons. And one of the things that we found was that if we did an additional drug screen, SUS12 deficiency or PRC2 loss results in sensitivity to certain epigenetic drugs like histone deacetylase inhibitors. So if you look at percent survival versus drug concentration in 3 types of isogenic cells, human Schwann cells, the ones that have the SUS12 knockout at a certain drug concentration just start to die. So this difference in sensitivity is not subtle. It's a very strong effect. The cells with PRC2 loss become dramatically sensitive to histone deacetylase inhibitors. And the reason that's important is histone deacetylase inhibitors are a drug class that are already FDA approved to treat T-cell leukemia. So there's 5 different HDAC inhibitors that are FDA approved. And the first one is called vorinostat. And we showed that it was active in the MPNST cell line xenograft in mice. If you look at percent survival versus days after treatment, in fact, it worked better than these other drugs that I told you about. So we did a simple-minded thing. So if you have one rock that can kill something, maybe you have a second one, you just try them together. So we combined a MEK inhibitor and an HDAC inhibitor. And it was great that what happened was the 2 drugs are synergistic. So the combination of the 2 drugs works better than either drug alone. And there's a formal way of measuring that called the combination index. The synergy was also observed in vivo. when we combined the drugs. In vivo, the two drugs were capable of shrinking tumors uniquely. Either drug alone could just cause the tumor to stop growing but not shrink it. And it was true with cell line xenografts as well as patient-derived xenografts. So this led to off-label use by Chris Mortel in a couple patients who had NF1 syndrome and PRC2 deficient tumors. This particular patient had unresectable brachial plexus, H3K27 trimethyl negative MPNST that you can see right here in this PET image. Radiation and chemotherapy had failed this patient already. And so Chris enrolled him on a combination of selumetinib and vorinostat. This particular tumor, because it's growing in a nerve, was painful. And this patient got pain resolution and a significant reduction in PET signal. And the combination was well tolerated. And this patient was on this drug combination for a couple years. This led us to seek a formal clinical trial, which is now open here at the University of Minnesota. It's a window of opportunity trial combining SpringWorks MEK inhibitor, and they're providing free drug for this mirdametinib plus the same HDAC inhibitor, vorinostat. Now, a window of opportunity trial means that after diagnosis or relapse, we're doing 28 days of treatment, and then the patient will get definitive therapy, whether it's surgery or radiation. So in that 28-day window, we have a chance using biopsies and imaging to see, are the drugs hitting the intended target? Is there a cellular response within the tumor to the treatment? Does imaging show potential activity? So the first patient is completed, and we're hoping to enroll second patient soon. Now, when we started this, Chris was running a trial, but now Robert Galvin, who's over here, has assumed leadership of the trial. So how did the first patient do? So this was a 27-year-old patient with the H3K27 trimethyl negative, axillary MPNST, treated neoadjuvantly for 28 days with this drug combination. And this is the PET imaging down here, pre-treatment and post-treatment. And you can see that the PET signal is dramatically reduced. So there was a 90% decrease in the metabolic tumor volume of the target lesion. A non-target lesions also decreased with lower SUV max. The MRI showed stable tumor size, but what you can see, this is the primary tumor here, is that it has markedly reduced contrast enhancement, suggesting reduced tumor viability, and that was seen in other lesions as well. So right now we're assessing the biopsies that we obtained pre and post treatment from this patient for phospho-ERK, H3K27 acetylation, which we expect to go up with HDAC inhibitor treatment, and Ki67 and markers of cell death. This drug combination was also well tolerated in the patient, both drugs taken every day. And also this patient saw the same significant improvement in pain that we saw with the other patient I mentioned. So we think about NF1 syndrome. It is a sarcoma predisposition syndrome. It could be a paradigm for thinking about prevention or early interception in cancer. We think that MEK inhibition can be potentiated by combining it with other drugs, and we found a bunch of candidates that might be useful in that regard. And the most exciting one is the HDAC inhibitor, where PRC2 deficient MPNSTs become sensitive to it, and there's synergy with MEK inhibition. So what we can think about in the future is that maybe some of these therapies will be useful treatments for MPNST, and you could imagine, if that's true, bringing them earlier in the course of disease as a way to intercept tumors early. So other researchers in the field have shown that if you look at an atypical neurofibroma by single cell sequencing technologies, within those atypical neurofibromas, there are the seeds of the MPNST there. So maybe we should be treating as early as possible when someone has that premalignant condition with some of these drug combinations. So we want to test the MEK inhibitor plus other drugs that I mentioned in a better genetically engineered mouse model using MRI readout, continue this window of opportunity trial, and plan potentially for a phase 1/2 trial in PRC2 deficient MPNST. I think the same drug combination would probably also make sense in other sarcomas that show RAS pathway activation plus PRC2 loss as well. So I'll stop there, and these are some of the supporters of this research. So certainly Rein sarcoma in the past, and others, and take any questions that you might have. So thank you.
This is really interesting. It raises, I mean, it's great work and it raises so many questions. So I don't know that we have time for you to answer all the questions that I have, but I'll start with a couple. One is the de novo incidence of this is really striking.
Oh, yeah.
You know, when Logan looked at TP53, there was only a couple or a handful of de novos, right? In your trials. That person of Austria, I mean, this is like 50%, right? So it's a lot.
Yeah.
Do you know, has anybody looked, is it, and I don't know, you can answer the question, is it because stochastically there's a lot of stuff going on in these early cells that are dividing at a rapid rate, and if you get a mutation in NF1, it's catastrophic? Or is there something peculiar about the organization of chromatin that makes NF1 and some of these other congenital syndromic mutations more likely to happen?
NF1 is super interesting. So it's a very large gene that's spread out over a huge genomic space, like 300 kb plus. So partly it's just a very mutable gene. It is also flanked by imperfect direct repeats. So some of the germline mutations are actually due to unequal crossing over between those repeats, and you get a deletion of the whole locus. And it's partly it just has a high mutation rate because it's so big. But also somebody, it might have been, you pointed out to me recently that when NF1 gets mutated in a spermatogonial stem cell, it actually gives that a selective advantage, and it proliferates, making more sperm that are NF1 mutant in this haploinsufficient state. So there's this, you know, if the mutation occurs, you know, that spermatogonial stem cell has a selective advantage, and you actually might get more, you know, it's more likely to get transmitted. So
is there paternal imprinting?
there isn't imprinting, but
Because with RB, there's clear paternal.
we do see there's an association between de novo NF1 and paternal age. So older men are more likely to have a child with a new case of NF1. So, yeah. Anyway, you know, it wouldn't surprise me that sporadic NF1 is also occurring in other species. I've always thought about we should be seeing dogs with NF1 over in the vet school, and so if we could figure out a way to easily look, it would be interesting.
The other question is, you know, this is clearly different from old age onset tumors, right? And so the work that De Gregori and Martin Carena have done independently recently tells that you have a lot of these events of clonal selection, but the environment sort of restrains. So you have these clonal populations that look like cancer at the intrinsic cell level, but the extrinsic environment just prevents them from becoming. Do you know if there's an extrinsic contribution? So if you look at the NF1 syndromic patients, is there any evidence of persistent CHIP or other areas where you're seeing these clonal expansions, and maybe they're constrained extrinsically?
Yeah, so I'm not sure if they've seen this in CHIP. But for sure, NF1 patients have patches of tissue that look normal, but they've actually lost the wild type allele. And it gives those cells a clonal dominance phenotype where they're more likely to take over space within the body. And then that could be the precursor to a benign or malignant tumor. So that definitely happens. So RAS and others have shown, like in model systems, RAS signaling can do that, where it gives a cell a clonal advantage. Yeah.
So can you think of ways that we can look at the extrinsic signals and maybe modulate the body's fitness or resistance to the tumor as opposed to trying to go after the cell intrinsic, which is really difficult because they're clearly unstable.
Yeah, people are trying to figure out ways to target the field effect. Yeah, but I have to think about that more. Logan?
Okay. All right. Yeah. Well, as you know, David, it is distinctly possible that in the next decade, we're going to have genomic newborn sequencing. So we will find these kids. I'm fighting hard to get NF1 and other cancer, childhood cancer predisposition genes onto the panel. And so what I'm wondering is, do you have like a time that you would give these things as preventive factors, or are you pursuing research or others pursuing research that can give you like good data on age at onset? You know, because I mean, maybe these kids are getting imaged and you can use an imaging or a cell-free DNA, something like that. But you also could put a lot of effort in, and there are a lot of computational tools now to try and figure out like age at onset.
For sure, people are doing that. So Brigitte Wiedemann and her colleagues at the NIH have this large natural history study where they're trying to follow NF1 patients with very advanced technologies, including imaging, as well as liquid biopsies to see, okay, when do these tumors really develop? When can we see the first signs of them? And that will provide useful data about potentially intervention or early interception. And some of the ideas that the field's thinking about are nutraceutical intervention. And so Chris Mortel and Pavlina Zverak here have done a year-long phase 1 trial of curcumin plus extra virgin olive oil to see if that nutraceutical intervention could be useful in preventing some of the symptoms of NF1. And they're actually seeing some significant effects. That was done in adults, but they'd like to move it to younger patients. And that has to do with the idea that there's data that inflammation is driving the formation of some of these benign tumors. And curcumin and extra virgin olive oil are anti-inflammatory and probably safe to give to children. So some of the challenge is what's safe to give to a developing child. The second idea is people are thinking about immunoprevention. Do these tumors express markers that could be recognized, using a vaccine? So you'd actually vaccinate the patient against their future tumor. So that's the other idea people have.
They don't need the microphone, but I was gonna say your idea about dogs, just like have a side story.
Yeah.
We had a sarcoma, soft tissue sarcoma in wolves, an endangered species. And I brought the ball of the tumor to the university, and Michael tried to do pathology with
really?
the vet school. This is a number of years ago, but we call the paper a clinical challenge because there's no antibodies for dogs to try to characterize PMNSDs or any sarcoma for that matter. So it's not that dogs probably don't have NF1.
Yeah, they do. We just didn't know. So it turns out dogs do get tumors that are called MPNSTs, and they're Schwann cell tumors. And Kyle Williams and I started collaborating with a group in Europe, and they sent some sections to us, and we started staining them, and we were very excited to see that a subset of them have high phospho-ERK and have lost H3K27 trimethyl. So dogs get PRC2 deficient MPNSTs just like human. Now we know that for the first time. I'm very excited about that because dogs might be a good way for us. It might be a good way for us to test some of the ideas that we talked about today, is actually treating these dogs, you know. And dogs get a lot of sarcoma. So, and wolves apparently. Is that what you said? It was a wolf? Okay. All right. Well, why don't we move on to our, oh,
Can I ask a quick question, Dave?
yeah.
Oh, sure. Nice work. I have a quick question. These two must have been shown to have mast cell infiltration that may be supporting their growth and survival. So in your drug screens, have you looked at combinations?
No. So in all the screens that I talked about today were monocultures of immortalized human Schwann cells with no mast cells present. Others have studied the mast cell. You know, as you point out, the mast cells are characteristic of the neurofibromas. They infiltrate them. And so mast cells can produce histamines. And so there's been clinical work with antihistamine treatment, but no convincing effect on the neurofibroma. The same with mouse models. They've tested the effect of mast cells on it, and they don't seem to make a big contribution to neurofibroma formation. In contrast, infiltrating T cells do seem to, and macrophage do seem to promote neurofibroma formation. So the mast cells might just be reactive somehow, and being drawn to the neurofibroma without actually, you know, causing it to progress.
Yeah, or is
All
it
right.
stage dependent phenomenon possibly at
Yeah.
different stages?
Yeah.
Thanks.
All right. Because I spoke so long, we're back on schedule. And so we can move to our next speaker. But because we didn't have any breaks planned in here, if you need to take a break, of course, feel free to. But we'll move on to our next speaker, who's Dr. Scott Schutze, clinical professor of medical oncology and internal medicine at the University of Michigan. Today, he will discuss leiomyosarcoma and the LMS spore. So please welcome me in joining. Please join me in welcoming Dr. Schutze.
Good morning or good afternoon. I guess it's afternoon for me, morning for you. And do I have the capability of sharing my slides?
Yes, we are ready for you to share.
Is it working?
Yep, you're all set. Thank you.
Okay, great. So thank you for inviting me. I have the privilege of providing sort of an overview and an update on the leiomyosarcoma spore. Just for background, SPORE stands for Specialized Programs of Research Excellence, and they were established in 1992 with really a goal of promoting collaborative and interdisciplinary translational research. And a key aspect of the SPORE is that not only is there an emphasis on the basic and translational research, but actually on clinical trials as well. So every SPORE is required to have at least one clinical trial, and each of the projects within a SPORE are required to have a clinical endpoint. And our leiomyosarcoma spore, which is generally these are funded for 5-year periods, began in September of 2022. And so the overall aim of our leiomyosarcoma spore is to translate laboratory findings into the clinic to improve the outcomes of sarcoma patients. And we have sort of 3 specific aims. One is to improve understanding of and exploit DNA damage repair vulnerabilities in leiomyosarcoma. The second is to uncover roles of germline DNA mutations in the gene TP53, but also in other DNA repair genes in the development of leiomyosarcoma. And then the third is to develop blood-based and also radiomic biomarkers for leiomyosarcoma. And our spore is a little bit unique. Many spores are just housed within one institution. Our spore is actually spread over many different institutions, over many different time zones within the United States, but we also have collaborators in Toronto, Canada, and we also have collaborators at the University of New South Wales in Australia. This sort of just shows a broad overview of our organizational structure. The spore is led by myself, who I'm a medical oncologist at the University of Michigan, and Jonathan Fletcher, who is a basic and translational researcher with a background in medical and pediatric oncology, who is at the Mass General Brigham. We sort of provide oversight of all the work that's done in the spore. We are supported by 3 different boards or groups. One is an external advisory board that has no institutional connection to the investigators that are participating in the spore. We also have an internal advisory board with representatives who aren't involved in the spore but come from the institutions that are involved in the spore. And then we have a group of research advocates. Within our spore, there are 3 translational projects. One is focused on genomic vulnerabilities in leiomyosarcoma. I'll go into each of these in more detail later slides. Second is focused on the role of TP53 in leiomyosarcoma. And the third project is focused on the development of biomarkers. And there is interrelation between each of these projects. And then the projects in the overall spore are supported by 3 cores, an administrative core, a biospecimen core, and a data analysis core. And I'll go into those a little bit more. And then finally, the spore also has 2 programs which are required. One is a developmental research program to fund some pilot projects that could evolve into further investigations that are supported by NCI funding, or that can become a full spore project. And then the career enhancement program, which seeks to fund, nurture, and develop young investigators in sarcoma research. So this is our external advisory board, composed of a number of current and past cancer center directors. We also have experts on the board in sarcoma and leiomyosarcoma more specifically, as well as rare cancer research. And we have a patient advocate, Maisie England from Sarcoma UK, who is also part of the board. Our internal advisory board is represented by investigators from many different disciplines. So we have representation from surgical oncology, from orthopedic oncology, from medical oncology, pediatric hematology oncology, cancer biology, and Mitch Ashe from the National Leiomyosarcoma Foundation is our patient advocate on the internal advisory board. And then we also have a specific LMS spore research advocate group, which is co-led by Mitch and Denise Renke at the University of Michigan. And we also have representation from Rein in Sarcoma with Michelle, the Sarcoma Foundation of America with Brandy, and an advocate at large, Taylor. So our spore is very fortunate, and we're very grateful for interest in what we're doing and commitment to helping support the work that we do. We have 2 families that have committed a sizable amount of money to help support our research. One is called the LMS 360 grant, and then we have a grant from the Susie Wolf Foundation. Both of these families were touched by leiomyosarcoma in different ways. We also have support from the National Leiomyosarcoma Foundation, from Sarcoma Foundation of America, and from Rein in Sarcoma. And we also have institutional support from Dana-Farber Cancer Institute, Mayo in Rochester, and the University of Michigan. So just briefly, we have three cores that support the spore work: an administrative core, which helps to manage all of the day-to-day activities of the spore. We have a data analysis core with leaders from the University of Michigan with expertise in bioinformatics and biostatistics, from the Mayo Clinic with expertise in clinical trials and biostatistics, and from the University of Toronto with expertise in large data management and bioinformatics that support the different projects within the core, as well as the developmental research program and the career enhancement program. And then we have a biospecimen core led by two experts, one at Mass General Brigham, who's a sarcoma pathologist, and a leader at the Nationwide Children's, who is an expert in biobanking and helps to manage biobanking of the biosamples that are collected as part of the spore work. We also have a number of interspore collaborations. So we're the leiomyosarcoma spore, but there are two other spores that are focused on research in sarcoma. There's a Memorial Sloan Kettering Cancer Center sarcoma spore, and recently, just this year, an MD Anderson bone sarcoma spore was awarded. There's also a radiosensitization spore that's at the University of Michigan that has some thematic overlap with the work that we do. And so these spores do collaborate together in a number of ways. We have a monthly sarcoma spore seminar series where we discuss or where investigators at our institutions present either their spore work or other work that is sarcoma related. Usually this is a work in progress, and it often isn't published work, but it just helps to provide an update on the work, the translational research, and the basic research that's going on around the country in different areas that have an interest in sarcoma research. We did host a mini symposium in March of 2024 at the Cold Spring Harbor Laboratories, and that was an opportunity for a number of investigators from some of these different spores to get together and interact. So moving on specifically to the projects that the spore is supporting, the first project is evaluating genomic vulnerabilities in leiomyosarcoma and ways to utilize those genomic vulnerabilities for therapy. This project is co-led by Jonathan Fletcher at Mass General Brigham and also by Suzanne George at Dana-Farber Cancer Institute. Each of the projects is required to have a basic research co-leader and a clinical research co-leader. And the three aims in this project are to characterize chromosomal instability, TP53 mutations, and DNA damage response in leiomyosarcoma clinical samples, to characterize DNA PK inhibitor response and resistance mechanisms in leiomyosarcoma, and to evaluate genotoxic therapies for leiomyosarcoma. So leiomyosarcoma is defined by chromosomal instability. There's a lot of genetic abnormalities within the leiomyosarcoma. A lot of it is not redundant, but there are some recurrent mutations or deletions in specific genes that are seen in leiomyosarcoma, which include TP53, RB1, ATRX, PTEN, as well as others. But this sort of chromosomal instability is a key feature in leiomyosarcoma, and it's always a bit of a mystery of how the cancer cells can maintain viability with all of this genetic disruption going on. And in a short hairprint RNA screen that was performed by Jonathan Fletcher's lab quite a number of years ago, they identified sort of two key genes as being sort of critically essential in maintaining viability of leiomyosarcoma. One is a gene PRKDC, which encodes a subunit of the enzyme DNA PK, which is involved in DNA damage repair, and another is RPA2, which is a gene that encodes a subunit involved in another protein that's involved in DNA damage repair and also maintenance of telomeres. And so the hypothesis in this project is that within leiomyosarcoma, there's this sort of intrinsic DNA damage that occurs, and there's defects in certain genes like TP53, RB, and others that result in sort of damage to the DNA and absence of repair of that damage. But there's enough, I guess, maintenance of DNA within these cells that viability persists. However, if you expose these cells, leiomyosarcoma cells, to some chemotherapy or agent that increases the amount of DNA damage, and then you block the ability of the leiomyosarcoma cells to repair that damage, that would sort of tip the cells over the edge, and they wouldn't be able to maintain viability. They wouldn't be able to survive in the presence of all of that additional damage and inability to repair that damage. And Dr. Fletcher's lab studied this in vitro in leiomyosarcoma cell line that was derived from a patient with metastatic uterine leiomyosarcoma, and basically showed that if you expose this cell line to very low doses of the chemotherapy drug doxorubicin, the cell's able to survive that very low dose, that very low exposure, and proliferate. If you, which is shown in the second second column. If you expose these cells to a inhibitor of the enzyme DNA-PK, which helps to repair DNA damage, the cells are still able to survive in the presence of this DNA-PK inhibitor. But if you combine the two, shown in the fourth column, the cells aren't able to maintain viability, and in fact, they die. They then modeled this in a patient-derived xenograft model, and using 3 different cell lines, leiomyosarcoma cell lines that they had, that showed that if you expose the mouse to doxorubicin or Doxil, the cells can continue to proliferate, as shown in the blue line, compared to no treatment is the black line. And if you expose the mice to the DNA-PK inhibitor peposertib, the cells can still proliferate, as shown in the green line, although less well than if they're not exposed to anything. However, if you give the mice a combination of doxorubicin and peposertib, then the cells are not able to proliferate in 2 out of the 3 cell lines, and the proliferation rate is much slower in the third line as compared to the others. So this concept sort of led to the idea of a clinical trial evaluating this concept. I'd just like to say that within their laboratory, within this project, there's a number of other enzymes that are involved in DNA repair that are currently being investigated to see sort of how critical they are, and if we can take advantage of inhibiting those enzymes to help improve the ability to treat patients with leiomyosarcoma. But based on this background, preclinical data, a phase 1 study was developed to explore the combination of this DNA-PK inhibitor, peposertib, which is an oral medication, combined with low doses of liposomal doxorubicin in patients with metastatic leiomyosarcoma, with the idea or the hypothesis being that peposertib combined with a low sensitizing dose of liposomal doxorubicin would be well tolerated and would demonstrate activity in leiomyosarcoma. This drug, peposertib, has been evaluated in combination with radiation, also in combination with full doses of chemotherapy, and has encountered significant toxicity when it's given sort of with full dose radiation or full dose chemotherapy. And so some of those other earlier studies were shown to really not be well tolerated for patients. The objectives of this trial are to determine the safety and tolerability of the combination, and then to determine the recommended phase 2 dose of the combination for further exploration, to assess the pharmacokinetics of the treatment, and to estimate the objective response rate and the progression-free survival in patients with leiomyosarcoma. And the accrual goal is 18 participants in the phase 1 portion, which is a dose escalation, and then another 12 participants in a dose expansion to get further data on people with metastatic leiomyosarcoma. And this is the study design of the trial. Participants have to be 18 years of age or older with inoperable or metastatic leiomyosarcoma. And in the dose escalation portion, we're also allowing enrollment of participants with myxofibrosarcoma, undifferentiated pleomorphic sarcoma, dedifferentiated liposarcoma, which share some similarities as far as the genetics go with leiomyosarcoma, and then synovial sarcoma is another histologic subtype that is eligible. At baseline, prior to treatment, tumor is obtained by biopsy and blood is obtained to look for cell-free DNA. After 1 week of therapy, tumors are biopsied again. This is to look to see if there's the anticipated changes that are occurring in the tumor cell, especially as it relates to DNA damage and DNA damage repair from the combined treatment. And then after the first 8 weeks, the tumor assessments are done with standard CT or MRI to assess tumor response. And this is the study design. Essentially, the starting dose of peposertib was at 100 milligrams orally twice daily, which is about half of the dose that is sort of usually given. And the dose of liposomal doxorubicin that was the initial dose was 10 milligrams per meter squared, which is about a quarter or 25% of the dose that's usually given as a single agent. And the study design was that initially the dose of peposertib would be increased up to 150 milligrams twice a day, then 200 milligrams twice a day. And then if at dose level 2, that full dose peposertib was tolerated with a quarter dose of the liposomal doxorubicin, then the dose of doxorubicin would be increased. And at the present time, we are at dose level 3, and it's estimated that around early April, we would have sort of the full safety data on this dose level 3 to determine if we will escalate to dose level 4 or if dose level 3 will be considered the recommended dose for the next phase of testing. Moving on to Project 2. This project seeks to understand and exploit the genetic basis of leiomyosarcoma development. And this project is co-led by David Thomas, who's a basic researcher, translational researcher, medical oncologist at University of New South Wales in Australia. And the clinical research leads for this are Judy Garber, who's has an expertise in TP53 and cancer and genetics associated with cancer predisposition, as well as breast cancer at the Dana-Farber Cancer Institute, and Josh Schiffman, who has expertise in TP53, who's a pediatric oncologist at the University of Utah. And the 3 aims of this project are to estimate the risk for cancers in families of patients with leiomyosarcoma and second malignancies in patients with leiomyosarcoma using the Utah Population Database and the Utah Cancer Registry, to examine the germline pathogenic variants in patients with leiomyosarcoma using the International Sarcoma Kindred Study, Count Me In leiomyosarcoma and leiomyosarcoma biomarker patients from Project 3. And aim 3 is to characterize the leiomyosarcoma risk in patients with pathogenic variants in TP53, otherwise known as Lee-Fraumeni syndrome. And this is through the Lift Up Consortium. So for the first aim, the investigators looked at the Utah Cancer Registry and abstracted out of the 200,000 patients with family records diagnosed between 1995 and 2021. They identified 450 patients or people within the cancer registry within the state of Utah who were diagnosed with leiomyosarcoma between 1995 and 2021. A few were excluded because we were unable to be absolutely certain of the diagnosis of leiomyosarcoma, which led 429 people identified. And these 429 people were sex and birth year matched with the population of people in the Utah population database that had family records and no history of leiomyosarcoma. And this sort of was the I guess, the data from which the risks of cancer were evaluated. And out of these two groups, the team found that there were 121 non-leiomyosarcoma cancers that were diagnosed in 89 of the 429 people with leiomyosarcoma in the cancer registry. And there were 409 non-leiomyosarcoma cancers in the 360 people with cancer in the population controls. And these graphs just show the relative distribution of the cancers, the non-leiomyosarcoma cancers, showing that breast cancer and prostate cancer were more common than other cancer types, and that's not unexpected. That's expected. But within the leiomyosarcoma families, melanoma seemed to be more represented than the controls, as well as colorectal cancer and hematologic, which ended up being mostly non-Hodgkin's lymphoma. And I won't go into all the details, but this research has identified that patients with leiomyosarcoma are at higher risk of developing a leaf-Fraumeni-associated other cancers versus the controls, and that patients with leiomyosarcoma have a higher risk of earlier onset cancer by about 10 years compared to the controls. And the relatives of people with leiomyosarcoma also have a higher risk of leaf-Fraumeni-associated tumors than controls, and also a higher risk of lymphoma, colorectal cancer, and melanoma. Briefly, there's a genome-wide association study that's looking for genetic determinants associated with the development of sarcoma. And this is looking at a very large database, the International Sarcoma Kindred Study, that's led by the Australian group, along with the UK United Kingdom group of participants, and using a very large controlled database of elderly people who are healthy and have not had serious chronic diseases or cancer. And this is really a search for novel germline genetic variants or mutations that might predispose people to developing sarcoma. And this is a work in progress. But looking at patients with leiomyosarcoma, looking at three different cohorts, looking at a cohort from the Dana-Farber, which is through the leiomyosarcoma Count Me In project, looking at the participants who are enrolling in the SPORE biomarker project, and then looking at people who have enrolled in the International Sarcoma Kindred Study that have leiomyosarcoma. We've identified a number of genetic mutations in the germline of these participants, and which are shown here, that likely have predisposed the people to developing leiomyosarcoma. And the risk within this population of having one of these germline variants that might have some direct impact on the reason why they develop leiomyosarcoma is about 10%, which is leading to the development of some recommendations and guidelines for recommending that patients with the diagnosis of leiomyosarcoma be referred for genetic evaluation, counseling, and testing. And then finally, for the third aim, they're capitalizing on this Lift Up project consortium, which is trying to understand and develop progress in leaf-Fraumeni syndrome, which is germline mutations in TP53. And within this consortium, there are 1,849 families that have enrolled. They have sort of verified that the families meet the definition of leaf-Fraumeni syndrome in over 1,600. They have germline data in the 1,300, and a total analytic set of 1,500 patients that are being analyzed. And what they've shown so far is that within the leaf-Fraumeni population, breast cancer is the most common cancer that develops, but sarcoma is the second most common cancer that develops, followed by brain cancer, lung cancer, and others, is shown here. And specifically looking at the leiomyosarcoma in this patient population, the age of onset is generally between between 30 and 50, which tends to be a bit younger than if patients don't have leishmaniasis, and that the patients with leiomyosarcoma often are affected by more than one cancers, either 2, 3, sometimes 4 or 5 different primary cancers that arise. Embedded within this is a population science project led by Wendy Coleman at the University of Utah and Mandy Ballinger at the University of New South Wales that are aiming to explore leiomyosarcoma patients' interest in and beliefs about genetic testing for predisposition using a validated tool and examine cross-cultural comparisons between the U.S. and Australia primarily, which is the International Sarcoma Kindred Study. And this is being done through questionnaires that are sent to participants. And this shows the sort of the approach for the questionnaires. The participants enrolled in the leiomyosarcoma spore biomarker study, which will be 200, are given a questionnaire to complete online or asked to complete a questionnaire online, and along with the participants enrolled in the International Sarcoma Kindred Study, and then participants that were identified at the Huntsman Cancer Center, as well as through outreach from the leiomyosarcoma patient foundations. So participants complete questionnaires online, and then if they have a genetic variant identified in their germline DNA through research or other means, then they're referred for genetic counseling and testing. They're asked to complete a questionnaire, and then the samples are sent off for clinical testing. And when the results are available and returned to the participants after that sort of genetic return of the genetic results, then patients are asked to complete another questionnaire. And this is just sort of preliminary, and this just shows the initial completion of the initial questionnaire. 366 have completed the initial questionnaire. The mean age of the participants is 58. Majority are women, majority are white. Most have some or more college education. The majority are married, and about 60% are from the US and 36% from Australia, and the remainder from mostly Europe. And moving into Project 3, which is applying liquid biopsy technologies to detect clinical response and mechanisms of resistance. This is led by Brian Crompton, who's a pediatric hematologist oncologist at the Dana-Farber, Brittany Ciantis, a medical oncologist at the Mayo, and myself, with the aims to determine the association of ctDNA levels and recurrent copy number alterations with outcome in patients with metastatic leiomyosarcoma. And aim 2 is to characterize and validate recurrent patterns of tumor heterogeneity and tumor evolution. And so looking at the first specific aims, which has to do with developing a biomarkers, we have an observational study in patients with leiomyosarcoma with a number of institutions that are participating. And this is for people who have metastatic leiomyosarcoma or inoperable localized leiomyosarcoma who will be undergoing first-line chemotherapy with doxorubicin-based or gemcitabine docetaxel regimen. And we're collecting, as shown here, collecting blood samples and sarcoma tumor samples, as well as collecting the MR and CT images at different time points along their treatment for up to 6 cycles, and then at the time of progression. And so far, we've enrolled 82 participants. It took a little while to get this off the ground at all of the different participating sites, but in the last year and a half, the enrollment rate has really picked up quite a bit. And this is just some very preliminary data from the first participant showing that we can detect circulating tumor DNA, as shown in the graph on the left, at baseline, and it persists on day 8 during cycle 1, but begins to then drop. So it's lower on day 1 of cycle 2, and eventually it sort of becomes undetectable down around cycle 4. And then after the patient's off of treatment, at the time of progression, which occurred a number of months after the patient had finished chemotherapy, we were able to detect the ctDNA again. And this is just a graph that just shows on the right-hand side for the patients with metastatic disease, we're able to detect circulating tumor DNA at baseline, which does drop over time in many patients, but not all of the patients. And we'll be eventually evaluating how the ctDNA change over time correlates with response to chemotherapy, progression-free survival, and overall survival in the patients. And just to finish up quickly, we have two programs. One is a career enhancement program, which seeks to provide funding for early career investigators. We provide $100,000 the first year, 50-50 with federal funds and philanthropic funds, and then it's eligible for renewal a second year with philanthropic funds. And we've awarded 6 different awards. We've awarded over the last few years. We've had awards go to investigators from a number of different institutions, not just the core SPOR institutions. So MD Anderson, Cleveland Clinic, Johns Hopkins, Texas Tech, in addition to Dana-Farber. And there's a variety of different areas of research that are being investigated by these investigators. With regards to the developmental research program, very similar, 50,000 in federal funds, 50,000 in philanthropic funds for the first year, eligible for renewal for a second year. And these are the awardees for the developmental research program. Again, it's not restricted just to people at the core SPOR institutions. We awarded Dr. Losa in the first year from Johns Hopkins, who's studying immunotherapeutic approaches to treating leiomyosarcoma. We're also investigating or supporting an investigator at Mass General Brigham, who's evaluating novel ways to disrupt the cell cycle progression in leiomyosarcoma. And we're funding some work at MD Anderson as well. So just quickly to finish up, our SPOR chase faces a number of challenges. One is that since we're spread across multiple time zones, meeting coordination has been a little bit of a challenge, but fortunately with Zoom, we've been able to work through many of that. We've also had some issues with interinstitutional data and material sharing arrangements, but have been able to develop a master agreement that we were able to get all of the institutions to sign off on to help facilitate the sharing of data and biospecimens. I'm not sure how much you're aware, but the executive orders from the current administration have significantly impacted our diversity efforts in the SPOR, as well as our ability to collaborate with our international colleagues. And then we've faced a number of issues regarding new regulations on multi-site participation in clinical research. And in conclusion, I just want to really thank Rein and Sarcoma for their support. The support that they provide does help to support not only our career enhancement and our developmental research program, but now with the new executive orders and not being able to use federal funds to support our international colleagues, we're using philanthropic funds in order to keep them involved in this project, in the SPOR program. So I'll finish there since I'm over time. And I'd be happy to answer any questions.
Any questions? Maybe I'll just have one. So has it been shown that ATRX mutations cause formal synthetic lethality with the DNA PK inhibitor? And how did that affect your inclusion criteria for the clinical trial you talked about?
So the clinical trial is enrolling primarily patients with leiomyosarcoma that all have, or pretty much all of them have mutations in TP53 or NRRB, and many of them also have mutations in ATRX. We aren't sort of pre-selecting patient enrollment based on the genetics of the tumor, but the genetics are being evaluated as part of the correlative studies that are being done. Does that answer the question?
All right. Yeah. Any questions online? All right. So let's thank our speaker, Dr. Kitsy.
Thank you.
Yeah. All right. Our final presenter is Dr. AJ Bakanaram here at the University of Minnesota. His presentation will focus on how CIC DUX4 sarcomas escape immune surveillance. So, thank you.
Good afternoon, everyone. Um, uh, my name is AJ, and today I'm going to present how CAC ducts for sarcomas escape the immune surveillance. This work was done at Darko Bosnopolski's laboratory, and I thank Rein Sarcoma for this opportunity to present here. So CIC ducts for sarcomas are highly aggressive, small round blue cell sarcomas of soft tissues. These are most common in children and young adults, and they present with high metastatic rate. They quickly develop resistance to chemotherapy, and the median survival rates in these sarcomas are way too low compared to any other um, uh, cancers in children at 5 years mark. So genetically, CIC ducts for sarcomas are a chromosomal translocation involving the translocation of CIC gene to um, DUX4 at chromosome 4 or 10 and forming a fusion oncogene, which from a transcriptional repressor turns into a activator, which involves key genes, activation of key genes in cell cycle regulation and oncogenesis. So the current challenges in CDS in basic research are unavailability of lot of unavailability of cell lines. We currently have, in my opinion, I think 3 to 4 cell lines currently, and those cell lines they lack the complexity of those primary tumors, and the xenograft models from those cell lines, um, they're immunocompromised, and the organoids are difficult to establish because of rarity of these sarcomas. So at in our lab, we wanted to model a genetically engineered mice model, which is which express CAC ducts for oncogene in a tissue specific fashion, and we wanted it to be an inducible model. So for that, we inserted the CIC ducts for oncogene in HRPT locus of embryonic stem cells, and then we transplanted them into the blastocyst of black 6 mice, and the chimeras were born. And when induced with doxycycline, these chimeras they develop soft tissue tumors under the skin, and you can see the histology that how they compare with the xenograft model of CDS, X1 and KITRA. And when we when we analyze the transcriptome of these tumors, they closely correlate with the xenograft model and biopsy samples from the patients. From this tumor, we derived we tried to establish a cell line which is doxycycline inducible, and when the doxycycline is removed, these cell lines is no more SCAC ducts for sarcoma cell line, and they undergo senescence. within 48 hours of removal of doxycycline and or shutting down the CIC-DUX4 oncogene. And these cells can be cultured in a sphere 3D model, and the target genes of CIC-DUX4 are highly relevant or highly expressed when the doxycycline is on, and when dox is off, these markers are just gone. One more important thing in having the cell lines is that when we establish the cell line, many of them they might not translate in vivo and form tumors. So to test that, we transplanted these cell lines in NSG mice, and we saw that these cell lines form tumors, and when the doxycycline is removed, the tumors shrink and they don't proliferate anymore or express any CDS target markers. And we wanted to see if this cell line has the potential to metastasize, which is one of the major problem in CDS, and patients are often diagnosed with the metastatic lesions on lung or liver at the beginning. So we injected the cells intratibial, and we saw that these cells metastasized to the lungs, and when injected intravenously, these cells metastasized to the home to liver and lung, which are two more major sites for metastasis in CDS patients. So in 2021, the work done at Darko Bosnikowski's laboratory, where he proved that CIC-DUX4 requires CBP 300 for its oncogenic activity, and when you inhibit the CBP 300 with IP300W, a potent P300 CBP inhibitor developed here at the University of Minnesota, and they saw that the cells no more. There is a absence of proliferation in vitro and in vivo both when treated with IP300W. And this inhibition was irreversible, and when these treated cells were chased for 7 days, they still don't come back to the proliferation, and they remain in that state. So we wanted to test this potential of CBP 300 inhibition in the cell lines we established, and we saw the same thing, that either when the CIC-DUX4 gene was off or treated with IP300W, in both cases they don't proliferate. So they need the CBP P300 axis for the proliferation. And we saw that also in vivo when treated with IP300W for 7 days. There was a significant reduction in the tumor volume. So, we analyze the microenvironment of the CDS tumors and compared with synovial and osteosarcoma, and we observe that CDS are cold tumors in terms of immune markers present in the tumor microenvironment. The variety of reasons for any cancer to exist as a cold tumor is either absence of antigen presentation machinery, or there is a immune suppressor cells, or the checkpoint regulation, or the microenvironment modulation through chemokines and other factors. So we went ahead to test whether this CIC-DUX4 sarcomas they have the ability to show the antigen presentation, and we saw that the CIC-DUX4 sarcomas does not express any MHC1 there's no antigen presentation. And when the DOX was off, which means the oncogenic activity of CAC DUX4 was off, the MHC1 resurface on their molecule, on the MHC1 molecule resurface. And this was even more profound when the stimulator interferon gamma was added to them. And a similar effect was seen by pharmacological inhibition of CIC DUX4 oncogenic activity, where we see the components of MHC1, H2DB, and beta-2M both resurface. So to test this in vivo, we injected these cells in a syngenic system where we did a time course study in which we shut down the CAC DUX4 oncogenic activity for 3, 6, and 9 days. And as we see that the CD45 population for the immune cells increase from day 3 to day 9. The macrophages population increases, and also there is an infiltration of CD8 positive T cells. Now we did the FACS analysis to see if the MHC1 presents itself after the inhibition of CAC DUX4. We see that from DOX on, where we have 11% of cells expressing this H2DB, to almost all the population expressing MHC1 at end of the study. And there was the infiltration of CD45 from 4 to 13% at the end of this study. We see that there was infiltration of the CD45 cells, and there is an increase in population of T cells marked by TCR beta from 4% to 35%, and there was increase in CD8 positive T cells from 3% to 27%, and there was a clear shift of T cells from CD4 to CD8 when the CAC DUX4 was absent. And there was the reduction in T regulatory cells, which were present initially when CAC DUX4 was on. And when the CAC DUX4 was off and the immune landscape shifts, the T regulatory cells are almost nil. And there was infiltration of this innate immune cells like macrophages, specifically this change in phenotype from pro-tumorigenic to anti-tumorigenic, marked by CD206 and F480, the macrophages shifts. And there was a increase in population of dendritic cells in tumor microenvironment. So from this, we hypothesized this mechanism. We proposed this mechanism that CAC DUX4 oncogene regulates the expression of MHC1 negatively, thus excluding them from presenting themselves to the immune system and presenting as a cold tumor. And the same it can be resurfaced when the oncogenic activity of CAC DUX4 was off, or either the cells were treated with IP300W, which again pharmacologically inhibits CAC DUX4 oncogene activity. So we know that hot tumors respond more effectively to those new age immune checkpoint inhibitors. The fight they're better in fighting the tumors in hot tumors rather than the cold tumors. In this case, CAC DUX4 sarcoma, it's desert where they don't present themselves, and thus there is no opportunity to exploit the immune checkpoint inhibitors for therapeutically challenging CAC DUX4 sarcomas. we did a pilot study in which we removed the doxycycline, inactivating the CAC ducts for sarcoma, the oncogene, and treating them with anti-PD-L1 to see if this immune checkpoint inhibitors fastens the process of tumor shrinkage. We see that there is a significant difference between the group treated with IgG and anti-PD-L1. Here you can see the histology of those tumors. So in conclusion, we successfully modeled a syngeneic mice for CAC ducts for sarcoma, and we demonstrate that oncogenic activity of CAC ducts for suppresses MHC1 antigen presentation to sustain the immune evasion. And our future directions will be to explore the potential of this CBP300 inhibition to re-express MHC1 antigen on CDS and exploiting that to make CDS vulnerable for immune checkpoint therapies. I thank all our lab members at Boston's lab and the funding agencies, Rein in Sarcoma, Children's Cancer Fund, NIH, and Minnesota Regenerative Medicine. Thank you, everybody.
Questions for Dr. Bhattamaram?
I wondered, you mentioned that the model transcriptome was similar to like patient-derived samples. I was wondering how many samples, how was this measured, how do you calculate these, and if this can be potentially further.
So the correlation analysis it was done by in the set of known CDS target genes, which we know previously. Yes.
And this came from like patient sample or?
So the publicly available database. I couldn't recall exactly the N, but it's publicly available database.
Any idea why it looks like just injecting IgG also shrunk the tumors? I'm just interpreting.
Sorry, we are really fast at that. So it was a Dox-off condition where the tumor was meant to shrink. So it was not. So we wanted to see the difference between the shrinking tumor with Dox-off and the one which gets a PD-L1, because the idea is that the CBP300 inhibition is similar to the Dox-off condition.
So the anti-PD-1 and PD-L.
This is, as I said, this is a pilot study, and we are working on this here.
Other questions? All right, if not, thanks again. All right, so just some final remarks. I want to thank everybody for joining us today, and also thanks to the presenters for their fantastic presentations. So we're going to break now for lunch, but also to visit the posters. Make sure to look at those and speak to the poster presenters. For those of you who are attending in person, the lunch is served in the back, so please feel free to help yourself. And we'll have time to network with colleagues and discuss what we heard today during the talks. Also, if you're interested, later, you'll have a chance to visit my lab, Kyle Williams' lab, and Robert Galvin's laboratories on the second floor of Malcolm Moose Tower. So if you're interested, let me know. The tour will begin maybe about 1:15 or so. We'll head over there if you're interested. So those of you attending virtually want to express our deepest thanks for being part of today's symposium also. We're grateful for your participation. I also want to remind everybody that Rein and Sarcoma has some resources over there on the table. Whether you're in need of information, support, or just want to learn more about the mission of Rein and Sarcoma, take a moment to pick up those materials that might be helpful to you. And once again, thanks for everybody for being here. And now we get to break for lunch. So Jimmy Jones. That's really cool. So you haven't shown yet if P300 inhibition can induce elusive platforms. It does. Also. So that's cool.
Can I put something on the screen here from the slides?
Yeah.
So let's see. It's good. So I do the right thing. Back to my
This one?
so hard in between transition, like I had
Yeah.
intros for people, but then
I know it is hard when they're not all. Nice.
I'll come back and change this like
Okay. So then this is there's probably a better way to do this, but next to see it. So then maybe redo this screen. Should be good here too. Sweet. Okay.